There's two of you in the class. Where are the others? So shall I start? Please ask others also to join. We can quickly go to the topic and then post the content in the Google Classroom, and from there you can go through. Shall I wait? Amreen, is anybody else also joining the class, or should I start?
Ma'am, begin, then they will join.
Okay. So we'll talk about the development of liver, pancreas, and extrahepatic biliary system. That is the gallbladder and the duct system of the liver and the gallbladder. Okay. This starts in the 4th week of intrauterine life, where you can see this is the endodermal side. This is the portion of GIT and the foregut and midgut. The junction of these two on the ventral side, that is on the endodermal side, develops a hepatic bud. And this hepatic bud grows into the ventral mesogastric. This is the ventral aspect and this is the dorsal aspect. So if you see, I'll tell you how. We go back to the previous. See, this portion. This is foregut. This one is midgut. Junction of this foregut and midgut is the site where the hepatic buds develops over here. Okay, and it pierces the septum transversum. This is septum transversum, right? So I'll move to the slides, and then you can easily understand how the things are developing. See, hepatic buds develop and it grows into the ventral mesogastrium to reach the septum transversum. Now this hepatic bud, before reaching the septum transversum, divides into two. Hepatic bud and a cystic bud. Cystic is responsible for the development of gallbladder and hepatic is for the liver. So here they've shown in the picture that this it could also be called as pars hepatica and pars cystica. So this hepatic bud is also known as pars hepatica, whereas the cystic bud is also known as pars cystica. Now, the division of pars hepatica takes place according to the lobes of the liver, and it is bifid, divides into two and further forms the right and left parts of the liver to make the complete liver. Okay. In the next picture, there's a colored diagram so that you can very well analyze. This is foregut, midgut, and the junction of these foregut and midgut on the endodermal side or on the ventral aspect gives rise to the hepatic bud, which further subdivides into pars hepatica and pars cystica, responsible for the development of liver and gallbladder, respectively. Right? Now, the part of the hepatic bud, this hepatic bud is there. It, before forming the liver, forms the bile duct that is responsible for the carriage of bile from the gallbladder. Via the cystic duct, it empties into the bile duct, and simultaneously there develop two more buds on the ventral as well as on the dorsal side of this developing gut. The ventral bud is responsible for the development of ventral part of the pancreas, and the dorsal bud is responsible for the development of dorsal part of the pancreas. And when the rotation takes place, that is, when the rotation of the midgut is taking place, this ventral bud along with the bile duct comes to lie below and behind this dorsal bud. We'll appreciate it in the next picture. See, this is the ventral pancreas, dorsal pancreas, and when the rotation is taking place, this bile duct and the ventral portion or the ventral pancreatic bud comes to lie below and behind the dorsal pancreatic bud, thereby developing the whole of the pancreas, which is in the C of the duodenum. So the ventral part of the pancreas or the ventral pancreatic bud is responsible for the formation of head and uncinate process, whereas the body and tail is formed by the dorsal pancreatic bud. Is it clear up till now? Can you hear me? Faiza, are you here? Okay. So it's quite easy. And these are the models which are present in the museum so that you can very well analyze how the things are there. See, this is the developing liver in the septum transversum from the hepatic bud. This is the cystic bud, and the ventral pancreatic bud is shown in blue color, whereas the dorsal pancreatic bud is shown in yellow color. This is the pink structure is the developing GIT. At the junction of the foregut and the midgut, this hepatic bud develops. And this is also the similar model. In this model, same colors are used for both. Now here you can see the development of the pancreas. At the junction of the foregut with the midgut, two buds are formed, that is the ventral pancreatic bud in blue and the dorsal pancreatic bud in yellow. With the growth of the gut wall and with the rotation, this bile duct along with the ventral pancreatic duct comes to lie below and behind the dorsal pancreatic bud, forming the pancreas as a whole. This ventral pancreatic bud is responsible for the formation of uncinate process and the head of the pancreas, whereas the body and tail is formed by the dorsal pancreatic bud. Okay. Now coming to the duct system of the pancreas. Duct system is in this manner. From the dorsal bud, a duct arises, that is the main pancreatic duct or the duct of Wilson. Then from the ventral pancreatic bud also another duct arises, that is known as accessory pancreatic duct of Santorini. And basically they have to form a communication between each other. In order to make the communication, what happens is the two ducts, they develop anastomosis. This ventral duct anastomosis with this through a transverse connection, and they form the main pancreatic duct whereas the remaining portion of the main pancreatic duct becomes the accessory pancreatic duct later on. When, see, this has fused with the anastomosis and this is left out. So this becomes the accessory pancreatic duct. Now coming to the anomalies of the liver, they are like there could be extralobation or there could be just a single lobe. Then there is Riddle's lobe. This is Riddle's lobe. Absence of quadrate lobe may also be there. Like when we will study the development, the cross anatomy of abdomen and pelvis, then it will be easier for you to understand. Also, there is accessory liver in the falciform ligament. Similarly, about the pancreas, pancreas could be divided, like dorsal and ventral have been fused. Then there could be accessory pancreatic tissue anywhere in the GIT, and the inversion of the pancreatic duct. That is, the main pancreatic duct is not formed through the union of ventral and dorsal ducts. Instead, it remains as such, and the embryonic arrangement is persisting. So that the minor duodenal papilla is responsible for the major drainage. And here, different anomalies of gallbladder are shown. This is agenesis, means absence of gallbladder. Pars cystica is not there. So, gallbladder is absent. There could be sessile gallbladder, that is, the stalk is missing. It's directly opening into the common bile duct. Phrygian cap could also be there. This is how the cap is there. This girl is wearing this cap. Similar is the case in the gallbladder, that you can see a folding to form a cap-like structure. This is Phrygian cap. Then gallbladder could be bilobe, double gallbladder. Then gallbladder may be present in the substance of the liver, that is intrahepatic gallbladder. It could be mobile or sessile gallbladder. And duct system may also be having anomalies in the form of bile duct may be absent, cystic duct may be absent. There could be non-canalization of bile duct or the cystic duct, and absence of hepatopancreatic ampulla. These are pictures to show you the sessile bladder, double bladder, then it is bilobed. There is a complete partition. Here there is complete partition, but outside it appears as one. Then there would be Phrygian cap over here, and gallbladder may be in the substance of the liver. So that's it about the liver and extrahepatic apparatus. I'll share this content in the classroom because the time is very less and we cannot complete everything here. So I have to go a bit fast also. So just 18 people are there in the class, although there were 45 earlier. Where have they gone? Yes, Vishesh, that's a complete PowerPoint. I'll share the development of GIT as well as extrahepatic biliary apparatus in the same classroom. Rather, I'm going to share it just now. I'll take the attendance. Acha, I'm not going to take the attendance. Meanwhile, you just fill up the Google form I'm sending in the WhatsApp group. Because Already there were 45 people and I can't leave them because they were also there in the class. Please fill this form today only, okay? Okay, you can leave class. Thank you.